So today I will be talking about my research on the neurobiology of speech and dance and the potential role of oxytocin enabling us for these two behaviors. Of course, both speech and dance are essential social communication behaviors that enable us, among other purposes, to communicate love, hence the relevance of this lecture to the topic of today's conversation. So, for most of the part of my career, I've been very intrigued by this important ability that we and other species share to be able to communicate through complex vocalizations. And there are several different ranges in this ability. If we were to map the species of the animal kingdom on a scala, according to their complexity, how complex the vocal signals that they use to communicate are, we would find in this top of the scala us humans along with parrots. And what really becomes interesting here is that not only are we humans and parrots the most advanced vocal learning species in the animal kingdom, we're also the only species of being able to entrain our movements, our muscle movements, to the beat in music. Or other words, we're able to dance. And so this overlap in these two important and complex motor abilities, on the one hand to speak or sing, and on the other hand to dance, is what led me and other colleagues hypothesize that, well, this overlap might not be a coincidence. It might be that actually these two important and complex behaviors co-evolved in a fashion, in a really intertwined fashion that we have not thought of before. So this truly inspired my research agenda to now study these two behaviors, speech and dance, in tandem, and study them together, starting from behavior, then down to the level of the brain pathways, to genes, and then tapping into all these findings that I will be showing to better understand how to propose therapeutic solutions for people that suffer from speech and overall body motor deficits. So what it is important to really highlight at the behavioral level is that both speech and dance are essentially motor behaviors. And this might be truly straightforward for dance because we all know and we can all see how we put into motion several of our muscles in order to dance, but it might be less straightforward for speech. The reason why is because we cannot directly see the most important rapid and rhythmic movements that are happening in real time as we're speaking. And I'm talking about the laryngeal muscle movements. But with the help of laryngoscopy, and you will be able here to see my own larynx actually, we're now able to see these really fast movements that are making us able to produce the complex vocalizations that we use for speech. So I guess that with this video, it becomes truly straightforward that both speech and dance are two motor behaviors. So this inspired the next question, which is, which are the brain pathways that are responsible for these complex rhythmic movements involved in speech and dance? And we studied first this question through functional magnetic resonance imaging, where we had subjects and analyzed data sets of people and their brain region activation while they were just speaking versus while they were producing rhythmic movements of several body parts while lying down in a scanner. And we focused this first analysis that I'm going to show to you here on the primary motor cortex, since this brain area is the central hub that controls a movement that is so important for both these behaviors. And long story short, what we are identifying here is that there are statistically significant overlaps of the brain regions that control the complex movements involved in speech and these other movements that are involved in dance. More specifically here, we're finding that in this yellow dorsal laryngeal motor area, there is a statistically significant overlap with the area that is responsible for hand rhythmic movements, such as, for example, in clapping with music. We found another statistically significant overlap in this more ventral laryngeal motor area that's devoted to speech motor control, and again, this blue hand rhythmic motor area. More surprisingly so, we found another overlap between this laryngeal motor area dorsally and this area that is responsible for the control of leg and hip rhythmic movements. This was pretty surprising because this might mean that potentially single neurons that are located in these brain regions where we found the overlaps might be projecting to the larynx in one context, for example, in a speech context, and might projecting in a different context, for example, in a dance context, to control rhythmic hand movements. Or other neurons up here, in a speech context, they might be enabling us to speak, or in another context, they might be projecting to knee muscles for us to dance to the music. Why this is important? Well, because traditionally in the field of motor neuroscience, what was believed for many, many years is that basically the primary motor cortex has a very specific organization where neurons that are located in this so-called arm area will only be projecting to one type of muscle, namely to the arm muscle. Or neurons that are located here in the laryngeal area, they should be projecting only to laryngeal controlling muscles. What we are finding here is that basically it is very possible that the primary motor cortex is much more flexible than we once thought. I will not speak about this in detail, but I also wanted to mention our current efforts into recording brain activity from individuals while actually dancing, standing and dancing, and not lying in an fMRI scanner. We made this possible through innovating on protocols that enabled us to run what we call here mobile or wireless electroencephalography. We were also able to break the record and actually record brain activity in individuals dancing, specifically 5 individuals dancing together, which was the highest record in scientific history, and the reason why we were featured in our favorite outlets, including in the New York Times. Now, going down to the level of the genes, right? Which are the genes that are responsible for these important motor behaviors that I'm talking about here today? Because if the control of these movements is to be found among other regions in the primary motor cortex, then what would be interesting is to find out if the primary motor cortex has a distinctive pattern of gene expression when compared to the rest of the brain. And when we really sought to clarify this question, what we found out was that there was a statistically significant upregulation, namely turning up of the gene expression of these specific genes that make possible the function of oxytocin, or in other words, the oxytocin gene pathway. And what was also very important is that the primary motor cortex, although it was the only area in the cortex where we found this really important upregulation, when looking subcortically, the brain area where we found this upregulation pattern of the oxytocin gene pathway was the brainstem, which is also involved in more direct motor control. And so one of my hypotheses is that it might be that oxytocin receptor containing neurons in the primary motor cortex might be projecting to relevant neurons in the brainstem to eventually control all these movements that I've been talking about in this lecture. Why is oxytocin important, and how can we interpret these findings in the context of the rest of the knowledge that we have about this system? Because, of course, traditionally, oxytocin was mostly known for its involvement in breastfeeding or in uterine contractions. But of course, there is more recent knowledge pointing to roles of oxytocin in more sensory behavior, such as, for example, in auditory perception or learning in humans and mice model species. But the link that remains to be found is whether indeed oxytocin might be involved in anything motor, such as the motor behaviors, motor control that we find in dance and speech. And to this question, what becomes really handy is my PhD work in songbirds, where I sought to identify a potential role of oxytocin in singing in these beautiful songbirds. Well, as we all know, research life is not so easy, because as soon as I started working on this issue, I actually realized that the very system that I wanted to study in songbirds was actually nowhere to be found, or at least the name oxytocin was nowhere to be found. In the same way that I could find oxytocin in all mammalian species and the oxytocin receptor, whenever I was trying to find out the oxytocin receptor in genomes of bird species, I was bumping into names like vasotocin receptor 3 or mesotocin receptor. And if we actually zoom out and we take a look at the rest of the vertebrate species, we will actually see that the landscape of gene nomenclature is equally or even more confusing. And this potentially is due to a very important underlying issue, which is that for many, many years, gene nomenclature has been based on function and sequence identity. Namely, two genes will be named with the same name if and only if they share the same function or a very, very high sequence identity. And this led to all these confusing and really variant nomenclature across species. What we found out and proposed was that actually there is a much more stable measure on which to base our gene nomenclature, which is synteny. What synteny refers to is the order of the genes, the one next to the other, embracing a gene of interest. So for example, here we have this mammalian human oxytocin receptor that is being preceded by some specific genes and followed by a specific order of some specific genes, which we call synteny. And this other gene here that for many, many years was called vasotocin receptor 3 is actually being embraced by the same genes. So what we proposed here is that based on this synteny, on this conservation of collinear order of genes around the gene of interest, this gene should also be named oxytocin receptor. This is the orthologous gene across species based on this conservation of the gene territory. And we did not only focus this analysis on the gene, on the 10 genes surrounding the gene of interest, we also went as far as 100 genes embracing the gene of interest, or even the entire chromosome where the genes of interest are located, and found evidence for this important conservation across vertebrates. And so all these results led us to propose a universal, unified vertebrate nomenclature where all of these genes will be having the same name across species. So with these orthologies resolved, I was now able to study the role of oxytocin in singing in songbirds, being sure that this was really oxytocin and not any other gene named mesotocin or with any other names that we see it goes by. And I was able to address this question using as a model species this wonderful songbird species being these zebra finches that can sing two different types of songs. One is a type of song that they just sing by themselves perched on a tree, called undirected singing. The other one being the type of song that they direct to a female in order to attract her. And scientists have long sought to identify differences between these types of songs, and have found out that when the male zebra finches sing to a female, they actually add several introductory notes before they start singing the main motif, something that they don't do when they sing this type of undirected singing. So we thought, okay, let's tap into this difference and inject an oxytocin antagonist and see what happens with these introductory notes. Of course, we had a control experiment where we just injected intranasally saline, and we found out that with saline, the birds kept singing with all of their erotic, fully fledged introductory notes. Instead, when we injected an oxytocin antagonist, we found out a statistically significant decrease in the number of introductory notes that they were producing before the main motif of their song. The decrease was so much that it made their song sound as if they were just singing to themselves. Namely, their song was really devoid of any erotic love features that the directed song has. Our next effort centered around asking whether oxytocin might be underlying differences in two songbird species whose song only differs in terms of its complexity. And here, evolution itself and nature gave us a great example to tap into. So the history of these two bird species goes as follows. These wild white-rumped munias, they were imported from China to Japan around 150 years ago. And apparently in Japan, they took great care of them. They used them as pets. They bred them in a very friendly way. They treated them really as pet friends. And as a result, after several generations, what they found out was that this strains of birds was actually much friendlier. It was not biting. It was not fearful. And importantly, it started singing a much more complex type of song. So if the domesticated type of song sounds like this, the wild precursor song sounded like that. So we wanted to ask if it is possible that oxytocin might be supporting any of these behavioral differences over such few generations. And so we genotyped the oxytocin gene in several of these individuals from the domesticated and the wild strain, and we found really very specific nucleotide differences in the oxytocin gene in spots of the genetic sequence where for the rest of the vertebrate species and birds in particular, we found a lot of stability and conservation. And we know, evolutionarily speaking, that conserved areas tend to be very, very functional, so that these very recent changes should have happened in the oxytocin gene for a reason. In terms of content of oxytocin expression in the diencephalon, we also found a statistically significant difference between the wild and the domesticated strain. All these differences that we're finding and that happen in such quick evolutionary times might be pointing to a role of oxytocin to these differences in behavior and specifically in singing that we see in these two bird populations. And then another very important question, well, we are talking about oxytocin. How about the sequence of the oxytocin genes? And specifically here, we asked the question about the receptors, oxytocin and vasopressin and vasopressin receptors in humans when compared to other non-human cousins, such as macaques, chimpanzees, bonobos, or even archaic extinct humans, such as Neanderthals and Denisovans. Well, what we found out was that there were specific single nucleotide polymorphisms, specific nucleotide changes that were either only present in both modern humans and archaic humans, but not present in any other non-human primate, or even specific spots in the genome, and here I'm showing some examples of the oxytocin receptor, that were only present in modern humans. And what we found about these locations where we found these polymorphisms was that these places were really important hotspots for social deficits, such as, for example, in autism spectrum disorders. We also found that these loci were located particularly in areas of open chromatin. And we know very well how important open chromatin is to bind other proteins and be involved in such important mechanisms such as gene transcription or DNA replication. Now, how do we leverage all this information to be able to propose important therapies for people that suffer from speech and overall whole body motor deficits? Well, one of the clinical trials that we have undergoing is one where we're trying to identify whether oxytocin and an intranasal administration of oxytocin in particular could help autistic children with deficits in the realm of speech and movement and rhythmic movement in particular. I really hope to be able to share these results with you as soon as possible. We're starting this study in February. We have some other studies that are now close to completion that I would like to touch on, where we're saying, okay, one way is to have these drug-based therapies, another way is to have behavioral arts-based therapies where we're trying to increase oxytocin naturally through behavior, right? And from the literature, we know that all the artistic behaviors, they really increase oxytocin. And based on the knowledge that we get from my studies here, we also know that actually when we're dancing, right, we are practicing several muscles, such as the hand muscles that I'm showing here, that actually depend from control of neurons that might be simultaneously also be controlling laryngeal muscles in other contexts. What this would mean is that by dance training, you might actually be tapping into the same neurons that might lead to speech improvement in people with speech deficits. And we're trying this out right now in people with Parkinson's disease, namely, could you get better at speaking by dancing? And so in this clinical trial that I'm going to present, it is one of the three that we are currently running. We had people with Parkinson's disease undergo a dance training for 12 weeks versus another group with Parkinson's that did for the same amount of time walking, right? And so what we're finding in resting state fMRI is that this area that we found that controls both hand and also laryngeal speech movements actually show differences when it comes to its connectivity with the precuneus. It had really stronger connectivity with the precuneus in the dance training group versus in the walking group. And we found that this was not only something that was identified in the brain, we also were able to see in just 12 weeks' time how the dancing group actually got better in their hand movements and their verbal fluency compared to the walking group. And so I really hope that in this lecture, I was able to highlight how important it might be to really study these two behaviors together, speech and dance. What we're learning is that at the level of the brain pathways, there might actually be partly overlapping brain regions in the primary motor cortex underlying the movement that is necessary for speech and dance-related muscles. At the level of the genes, we're identifying that the oxytocin gene pathway family is highly upregulated in the human primary motor cortex and brainstem. It is a family that is really highly conserved across vertebrates. We can really use it for translational studies and be using the same gene names to refer to the same system across vertebrates. And we found through functional experiments that it is really necessary. Oxytocin is very necessary for specific bird-shot courtship features, and that it is differentially expressed in vocal learning pathways of avian species that just differ in song complexity. And we also were able to reveal some modern human-specific changes on the oxytocin receptor among other genes of the family. Right now, at the level of the therapy, we have an ongoing trial where we're trying to identify potential role of oxytocin in alleviating speech and overall whole body motor deficits in autism. And also we're having these very intriguing dance interventions in Parkinson's disease, where we are testing everything from fMRI connectivity to behavioral outcomes. Now, when it comes to what does this all say about the origins of humanity, the origins of love, I think at a very basic level, what all this research can tell us is that, well, we might actually be using a very flexible motor system in order to control the muscles that are responsible for important behaviors that enable us to express love, such as speech and dance. More evolutionarily speaking, it might be actually that once you get the ability to control in a very complex and rhythmic and rapid fashion one type of muscle movement, such as the laryngeal muscle movements, you might be actually buying directly the same rapid and complex and rhythmic motor control of other muscle movements, such as arm, legs, and other muscle movements that are involved in dancing. So really, dance and speech evolution might be much more intertwined than what we once thought. And we were not the only species where we see this intertwined coevolution. We also see it, as of now at least, in parts. So we're not alone in this journey. There might be other convergent journeys happening in the animal kingdom where we see possible similar trajectories. I also want to highlight that in my own journey, this involvement of dance in my research has actually made me put both my hats of a researcher and a dancer in the laboratory and be able to imbue my flamenco dance expertise in protocols that we're running for people with Parkinson's disease. So it's a very exciting moment for me to be really combining these two parts of me in the laboratory. I really want to thank all the members of the team that have been running so wonderfully this analysis. I couldn't appreciate their work more. And thank all the leadership at Rockefeller for making this work possible. Of course, all my collaborators and everyone who one way or another is supporting this research. And thank you so much for your attention.